Okay, hey Biochem, this is the visualizing proteins video. After watching this video, you should be able to describe the different renderings for a macromolecule. So this doesn't have to be proteins. It could be sugar, it could be nucleic acid, it could be lipid. It all counts. List a use for each of the different renderings and understand the qualities of a good figure. Okay, we're going to start with a ribbon diagram. So ribbon diagram, which is this view right here, super popular in biochemistry textbooks, well, all sort of biology and chemistry textbooks. And I'm going to tell you a little bit about what it means, because I'm not sure. I mean, I'm sure you notice it, and you notice it's blobby and it's colorful, so therefore it must be a protein. But it actually does have a meaning. So we'll start with the very, very, very basics. The ribbon traces the pathway of only the backbone. So that's why you can see the secondary structure, because the secondary structure occurs only along the backbone. There's no side chains here whatsoever. And the sheets, or the strands of a beta sheet, are arrows, and hopefully you can see this. They always point towards the C terminus. So here I have the N terminus labeled, and if you could trace this along from N to C. So this beta strand is connected to this alpha helix, doop, 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 which is connected to a 310 turn, which is connected to this other strand. These two sheets together, or these two strands, equal a beta sheet. And hopefully you can see that they're parallel because they're pointing in the same direction. This is connected to another 310 turn and another small alpha helix. And the reason that I know these are alpha helices or 310 turns is because it turns out this protein is one of mine. I solved the structure of it, so that's how I know. Let's see, the other thing that you should understand about this particular view is that the colors also go in order of the electromagnetic spectrum. So you could follow this from N to C, all the way from the colors through Roy G. Biv, only it goes backwards in Roy G. Biv, so blue to red. So blue is where nitrogen starts, and red is where the carbon is, and I wonder if you know why that is, because nitrogen is blue, right? And the carboxylic acid C terminus down here has oxygens on it, so red. So like I said, this is a really common view. It's not my most favorite view. So the ribbon diagram is sort of analogous to a person's, you can get close to that one, skeletal structure. So really all that you see here are the bones. You don't see all the good stuff like the tissues and all the atoms that fill in the space. And this gives students a sense that the protein is mostly empty space because you can see through it, and that's just not true. It is so packed with atoms, it's so packed with electrons that you can't see through it. So if we were to show all the side chains and all the atoms and drape a sheet over it, we get a much more accurate understanding of what the protein looks like. But this is the simplest view. It's simple, it's easy to look at, and it shows secondary structure. So that's why people like to show it. Okay, so here are, oh man, the formatting got off on this. I think you can figure it out. So here's all the other different renderings that you'll be able to pick from once you get into UCSF Chimera. You can download it now if you want to. It's free. You don't have to. I'm going to go through each of these. So again, this is a protein that I've solved the structure of. This is a ribbon diagram. Hopefully you can see that this is a trimer. There are 3 subunits, and they're all each different colors. Again, the ribbon diagram is useful for showing secondary structure. And in UCSF Chimera, you can also use what's called pipes and planks. It's the same thing. The planks are the beta sheets and the pipes are the helices. And this also is just a different way of showing secondary structure, but it's a little bit more, the secondary structure is a little bit thicker, so it's getting closer to representing the actual space that the atoms take up. Food for thought. Surface don't know where the E went. Surface here is my absolute favorite one. This is the best view, I think, for determining or predicting a protein's cellular location. So what you're seeing is CPK coloring, where the nitrogens are blue, oxygens are red. If they're nice and evenly, if the blue and the red is evenly distributed, then you're looking at something that is water soluble or hydrophilic, meaning that it's probably located in the cytoplasm or a cytoplasm of some organelle. So it could be in the matrix of a mitochondria, it could be in the cytoplasm of the cell itself. The other thing that you can see in a surface is you can find pockets where the active site might be, active/binding site. Maybe I should just call them like the functional sites. The functional location. So proteins have lots of functions, and the entire protein is actually not functional. Most of it is just scaffolding. But there are some parts of it that are functional, and those tend to be sort of cavernous cave structures or river structures. I don't actually have a good view of the active site in this one, but food for thought. Okay, spheres has a lot of the same functions that surface does. So it's good, again, at predicting cellular location. It is not good. It won't help you find pockets very easily. What spheres are the space that the atom takes up, or the space the electrons take up. The space the electrons take up. So here is the view that you really start to see that the protein is stuffed full. It's packed. It's not empty space like the ribbon diagram and the pipes and the planks make you think that it's, not very packed in with atoms, but spheres helps you see that it is super packed with atoms. They're all tightly packed in this really teeny tiny space. And again, spheres here in CPK coloring can help you determine the cellular location. If there were like large patches of gray, large patch of gray, that would give you an indication that the protein is membrane bound. Those are really, at this point, your two options. Whether it has some hydrophobic patches to it, means that it will be associated with a membrane. You don't know which membrane. Could be the ER, could be the cytoplasmic membrane, could be the nuclear membrane, could be the inner or outer membrane of the mitochondria. You don't know, but it would just potentially be membrane bound. Whereas if the red and the blue are evenly distributed, that gives you an indication that it's water soluble and therefore in the cytoplasm or inside the matrix of the mitochondria. So these three down here, ball and stick, and wire, are not typically the best views to show an entire protein. As you can see, it's super messy and super convoluted. Ball and stick is exactly what it means. I'll just zoom in here. All the balls here are atoms, and all the sticks are the bonds. You really, in a protein, you probably would show most of it as a ribbon, and then select the amino acid side chains that you wanted to highlight and put those in ball and stick, because it can help you show. The nice thing about ball and stick and wire is it shows you the orientation of the side chains. But usually you want to have your protein in mostly ribbon, and then select the side chains that you want and show them in one of these three. And that way you can show the orientation of the atoms relative to one another. Sticks is essentially ball and stick, except for the atoms are now stick instead of balls. And again, you can see that they're all still CPK colored. If you saw that this was mostly purple, it turns out that that's just the carbon atom that I've changed to purple, but here it's gray. And then wires, my least favorite. Oops, why do I keep doing that? Okay, so wire's my least favorite. It's the hardest to see because it's just super, super thin. When you open Chimera, most things will automatically go into wire. I do not know why. The highlighting in green out here is because I've selected the entire thing so that you can see it just a little bit better. I suggest that when you learn Chimera, you highlight everything that you can and move it out of wire. It's just really hard to work with. But essentially, it's just a thinner stick version. Okay. So those are the different rendering types that you have to pick from. Again, you could render an RNA, a DNA, a lipid, a sugar, doing all of these same renderings. You just have to pick which one you think suits your purpose the best. So that kind of leads me into figures. So making figures. So this is for the molecular modeling 2 assignment, well 2, 3, 4, and 5. You'll have to make figures, and I wanted to give you a heads up about what makes a nice figure. So for us, in this assignment, and when you look in papers, you'll usually see that the background is white. You'll also notice that almost everything that's extraneous is hidden. Extraneous, whoa, that is how I spell extraneous. Extraneous is hidden. So you really only want to show what your story is trying to tell, telling. So for here, I'm trying to demonstrate that there's a hydrogen bond in between these 2 amino acids. So I just have those 2 amino acids highlighted. I have the hydrogen bond between them. What I don't like is there's no labeling here. There's no label. You can't easily tell what these 2 amino acids are. There's also no figure caption. Please read your directions on how to make a nice figure caption. What else don't I like about this? Yeah, mostly it's just the labeling and the figure captions. Nice white background, no extraneous information. It's cleaned up. It's not pixelated. Again, the same thing with this one. Nice white background. There's a little bit of extraneous stuff here and here and here because I feel like whoever made this figure was trying to demonstrate that this green piece hydrogen bonds with that residue, which means you don't need all the extra fluff. Again, it's pretty much cleaned up. It's not too hard to look at, but there are some extraneous pieces. I'm not sure why that's there. So think really hard about what do you want your figure to say, and work hard at getting it nice and cleaned up so that it says that. Otherwise, again, this needs labeling. So label, what is this? What is that residue? And then it also needs a caption. I suggest you put all of your images into PowerPoint and use text boxes and arrows to label things and add your figure captions into PowerPoint and just submit your document for assignment 2, 3, 4, and 5. All as PowerPoint documents. And I think that's all I have for visualizing proteins. I'll see you in class. Bye.